We're lucky to welcome Dr. Crary today. His program focuses on defining neuropathological changes in human brain tissue, translating these observations to the laboratory and ultimately back to the clinic. His laboratory has proven success at producing high-impact interdisciplinary translational research related to both characterizing novel histopathological phenotypes in patients with neurodegenerative disorders, as well as modeling these changes in transgenic animals and in vitro cellular models. He led a project to perform a detailed study on the molecular changes occurring in the brains of patients with primary age-related tauopathy and spearheaded an international effort to define and provide consensus criteria for the entity. This project led to the identification of JADE1, I hope that's pronounced that way, as a candidate modulator of tau toxicity. These projects were all published in Acta Neuropathologica, the neuropathology journal with the highest impact factor. He also recently contributed to efforts to define and establish neuropathological criteria for other neurodegenerative diseases, including progressive supranuclear palsy, age-related tauopathy of astroglia, and CTE. He's developing animal and cellular models for neurodegeneration to advance our understanding of other molecular drivers of neurodegeneration. For instance, he has identified a neuronal microRNA termed MER219 that is dysregulated in Alzheimer's disease and in part, and it silences the tau mRNA on the post-transcriptional level in vivo and in vitro. These findings were also published, but now in the Journal of Clinical Investigation. Moving forward, Dr. Crary is continuing to enhance and expand his program, looking at the neuropathology of neurodegenerative diseases. The current focus is to leverage deep machine learning and digital neuropathology to identify new morphological traits that can be leveraged for clinical pathological, genetic, and other studies. He currently has NIH and foundation support. We've invited him to teach us about trauma, and so welcome.
Dr. Martinez-Lage, thank you so much for that introduction. I very much appreciate it. And thanks to all 49 of you for joining this Zoom call today. It's too bad that I can't see you all in person and see your faces and all your shocked reactions to the things that I say. But please, yeah, definitely put questions into the chat and I'll do my best to answer them. Yeah, I've really just been blessed to be in neuropathology research. It's so lucky that we get to do what we do. And working with human tissue, being able to do our projects and see what's really going on, and then try to come up with mechanisms and model them. And it's really high impact and very important. So for everybody who's here on the call, what you're doing is really important, and keep up the good work. All right, so here's my presentation. So just to give you like a little background about me, I'm a neuroscientist. I did a PhD in long-term memory, and then later I became a neuropathologist, and I just really got excited about trying to figure out, what are the neural substrates for all these really interesting things that I wanted to learn about. But my lab is mostly focused on the long-term effects of traumatic brain injury. But I put together a presentation that incorporates a bunch of review on kind of acute traumatic brain injury. And then I've also kind of sprinkled in a little bit of AI and deep learning, because I know that you're all kind of excited about that. I have nothing to disclose. So I'll give a background on acute, presentation on acute TBI, and then move into chronic TBI. So I know it's really important to think about definitions in pathology. So what does it actually mean to have a traumatic brain injury? So this definition came from this paper by Menon et al., but there's lots of ways that you can define it, lots of different causes. But essentially, it's an alteration in brain function caused by an external force that's applied to the brain. The causes are innumerable. Some are penetrating and some are non-penetrating. The trigger, which it's different from Alzheimer's disease, which we know what happens kind of at the end, but we know very little about the beginning. Traumatic brain injury, we know exactly what the trigger is. It's a mechanical deformation of the brain parenchyma. And then mayhem ensues, basically. Triggers a complex downstream cascade of events, very complex, can involve multiple organs. It's dynamic, and it can vary. So when you think about TBI, it's really important to take kind of a holistic approach, I guess, with everything in neuropathology, macroscopic, microscopic, and molecular. And even the clinical is, as you all know, is really important as well. It's also really important to think about whether an injury is mild, moderate, or severe, focal or diffuse, acute, or chronic. This is a classic and very important figure with some modifications here, Giza et al., 2001, which attempts to kind of put this complex cascade of events into a single graph. So initially, right after the TBI, there's a surge in calcium and potassium. Glutamate also goes up, as does lactate. There's differences in, or sorry, increase in the glucose metabolic rate as the cerebral blood flow goes down. So these are kind of the first events. After many hours, there's an inflammatory response that gets triggered. There's axonal injury, shearing, compression, and tearing of axons. The pituitary can be involved. It's only until many months or years later that this degenerative phenomenon starts to happen where tau and amyloid accumulate. So that is actually really important when we think about CTE and mechanisms, this idea of a latency period. Why is it that people get concussions and then seem to recover ostensibly, and it isn't until years later or decades later that they start to get symptomatic from that? Anyway, something to think about. So here's another important concept. So human data, as you can imagine, is super important when understanding traumatic brain injury. You could try to model things in mice and rats and other animals. Just an aside that I didn't put into the presentation, there's something called the scaling factor, which is really, really important. It's a mechanical phenomenon where things that are smaller are stronger proportionally to things that are larger. So you can imagine if a mouse fell off of a bookcase, it would survive that no problem and kind of scurry away. But if proportionally like a human fell off of a bookcase, to scale up the size of the bookcase, it would be splat. You can't pick up a whale by its tail because it would just tear in half. If you pick up a mouse by its tail, you can literally twirl it and it's just fine. So that's because of this, and it's because of the scaling factor. So you really have to work with a human brain, human specimen that's the actual proper size, the proper geometry in order to understand the mechanics there. So a lot of people have looked at severe and mild TBI, moderate TBI. This is a very famous curve, the Wayne State tolerance curve, where they plot the G's, basically the measure of force of the traumatic brain injury against the duration of time that the force is applied. And this is actually kind of built not from a single experiment, but it was multiple experiments in different contexts that were able to produce this. But the point is that if you can apply a strong force, but if it's just a small amount of time, the brain can do okay. And anything kind of above the curve here, above the tolerance level, will result in potentially severe injury, and anything below is probably okay. All right, another important thing to be cognizant of is this Glasgow Coma Scale. So this was built, clinically. Teasdale, I put the reference for you, 1974. The idea was there a way to kind of predict clinical outcomes after a traumatic brain injury? So they put together a score based on eye opening, verbal responses, motor responses. It's a semi-quantitative scale, and you can sum all these things up together. And then you can kind of predict after a TBI the probability of a favorable outcome, probability of death. So you're going to hear a lot about the Glasgow Coma Scale in various contexts. The problem with the Glasgow Coma Scale is that traumatic brain injuries are complex. They can involve different parts of the brain. And so maybe a mild injury in a critical region would be much more significant than, say, maybe a more severe injury in the, say, a less important region, maybe the right frontal lobe, for example. So this is really important to keep in mind. So more recent studies focus on kind of more the anatomical distribution and the pathology. So when you think about TBI really important to break it up into mild, moderate, and severe. And this is how it's operationally defined by many. So a mild traumatic brain injury can be thought of as a concussion. That's a term that is fraught with problems. Anyway, the classic definition of a concussion, there'll be no structural changes on an MRI or any kind of neuroimaging. Whereas in a moderate or severe, you may have an abnormal scan. Loss of consciousness, less than 30 minutes for mild, 30 to 24 hours for moderate, or more than 24 hours. Loss of consciousness would be severe. Alteration in consciousness, a moment up to 24 hours might be considered mild, more than 24 hours based on other criteria. Amnesia is important, a day or up to over 7 days for severe, and then this Glasgow Coma Scale that I mentioned before. So kinematics. Really important to understand the different ways that the brain can be damaged. So kinematics is the branch of mechanics concerned with like the motion of objects. Doesn't really matter what the cause of the injury is. So this is kind of more of a physical, mechanical discipline. So yeah, I guess what's really important for neuropathology is that there's certain types of forces that are maybe more important than others. Think about the kind of a linear acceleration that might be applied to the brain when it's kind of in the midline or from one side versus like rotational acceleration when the brain is forced to like spin around. Think of a bowl of porridge. These rotational injuries are really, really damaging. And that's because they lead to shearing forces. So in this image, you can see there's a little square here. And when you rotate the bowl of porridge, you form these, I guess you kind of stretch the material here is showing these like lines that tears the tissue. So this rotational injury is probably more important than the linear acceleration forces. The linear stuff is easier to model. It's kind of easier to understand. The rotational stuff is more complicated, but thought to be more important. So if you're going to get punched, you don't want to get punched in the jaw and have your head spin. These are like the knockout blows that will often happen in a fight. The linear acceleration from a straight-on blow is safer. So another important concept is stress concentration. So what is that? Well, I guess you can kind of understand it as somebody lying on a bed of nails. If the weight or the forces are distributed across a lot of points, you're not going to go through that nail. But if you're lying on a single nail, all of that force is going to be concentrated on that one nail, and that's going to lead to a puncture and so on. So if you want to reduce stress, you need to increase the area that the forces are distributed. As you walk around the world, you might start to see cracks in the pavement, like I'm showing here. Obviously, this brick, all the stress is getting concentrated on this one little point. That's why that crack formed there. It didn't form in a random place. Here's a candy wrapper. So you got a tear right at the bottom of this little nook tear here. When if this was not serrated like this, it's really hard to open. But having these little serrations there allows you to focus the stress at that one little point. So yeah, these are important. And the strain, that's like a measure of the deformation of the material. The material can fail when the stress exceeds the maximal strength. So, all right, so let's talk about some neuropathology here. So, acute TBI. In acute TBI, these focal lesions are really important. Hematomas, contusions. So, the hematomas, we'll go into those a little bit more deeply. They can be subdural, epidural, intraparenchymal. These are commonly tested on the boards. They're generally lead to local damage from reduced blood flow, and then that triggers ischemia, edema, and everything that goes along with that. Contusions are kind of bruises of the brain. They're hemorrhages at the site of impact, really where the brain comes up against bone, the bony prominences, common in the frontal and the temporal lobe. Typically, the crests of the gyri, the poles, the temporal pole, frontal pole, occipital pole. The TBI, the lesion can be on the opposite side or on the same side as the injury. Call that coup if it's on the same side, counter coup if it's the opposite side. The hemorrhages from contusions as well, as I mentioned before, ischemia, edema, necrosis. Long term, after weeks to months, we're going to get cavitation and gliosis. Clinically, this stuff can be diagnosed on a CT scan, blood. There's blood biomarkers, GFAP, and other brain proteins become elevated relatively quickly and stay elevated for a long time. So here's an example of an epidural hematoma on the right side of this patient's brain. You can see here's the dura, here's the brain, and a large hematoma, fresh hematoma here. These come from tearing of the meningeal arteries. The meningeal arteries are on the surface of the brain. Here's a trichrome stain. Here's the meningeal layer here. There's this dural sinus. Embedded within that are going to be these meningeal arteries. This is the periosteal layer of cells on top of that. Here's an example of a subdural hematoma. These may have a kind of crescent appearance on neuroimaging. And you can see this is on the right side of another brain, extensive from the frontal lobe all the way to the occipital lobe. After time, you can get a chronic subdural where the blood becomes resorbed and granulation tissue forms. It can be very vascular, and then it can rebleed on top of itself. You should be able to peel this membrane off of the dura. Here's an example of a subdural membrane getting peeled off the inner table of the dura. Here's a trichrome stain showing what this membrane looks like under the microscope. You can see these giant venous structures here that can rebleed. Here's actually a fresh hemorrhage here. So this might be a chronic subdural that then has rebled. Here's some examples of some contusions. So I was mentioning anywhere where there's contact of the brain with these bony prominences. So here's the temporal lobe bilaterally, and you can see hemorrhages here that are in the subpial space and potentially go into the parenchyma. The orbital frontal gyri is also a classic place for these contusions. And we're also seeing here on the frontal pole. Over time, these contusions get resorbed. You're going to find discoloration of the brain with an orange, kind of rust-colored hemosiderin staining or hemosiderin discoloration. You're going to see atrophy and cavitation, encephalomalacia, and so forth in a chronic contusion. Here's a coronal section through a brain that had undergone a severe TBI acutely, showing a lot of different features that you may encounter. So here we're seeing hemorrhages in the orbital frontal gyri. This is the interface here. Here there was a shear, so you get these like hemorrhagic tissue tears here in the corpus callosum, and we're seeing one here, another hemorrhagic tissue tear that comes from a rotational injury. Here's another one here. You can see midline shift potentially. Here are some petechial hemorrhages in the basal ganglia. So, so there's going to be these kind of focal lesions, but then there's also kind of diffuse injury that you also see. This can be seen in mild to severe TBI as well. So we're adding in the mild here for diffuse injury. So this is a much more widespread distribution. There's two major types. There's diffuse axonal injury and diffuse vascular injury. You generally don't see them apart. They generally occur together. What happens? The shearing forces disrupt the axons. Axons are really vulnerable to traumatic brain injury. They're long, they're thin and delicate. So when they get stretched, they can undergo an axonomy, like a primary axonomy, just physical, or they can just be stretched and then it damages the cytoskeletal components and disrupts transport, and you get these kind of spheroids that form, which is the same morphological structure that happens whenever fast axonal transport is disrupted. The axons can degenerate, and then you can sometimes get a secondary axonomy. If that happens, then you'll get Wallerian degeneration. The microvasculature is also affected. It's more resistant than axons, and you may have multiple small hemorrhages throughout the brain. People who have repetitive mild traumatic brain injury, we're going to talk about CT in a bit, can also have a kind of vasculopathy that resembles arteriolosclerosis, and you may find lots of hemorrhages around blood vessels in that context as well. So what happens, as I mentioned, is axons are super vulnerable to diffuse axonal injury, triggers this constellation of changes. So the stretch of the axonal lemma, the microtubules and neurofilaments get damaged. The membranes then get damaged, and then you'll get this metabolic, a triggering of this metabolic cascade. There's altered permeability, calcium dysregulation from internal stores, and externally, the axons become impaired as axonal transport gets impaired, and then you can get either sublethal or lethal cellular change, the secondary axonomy. So excitotoxicity is really important, and as is Wallerian degeneration. Excitotoxicity is a cell death mechanism, which is essentially restricted to the brain. This is just the glutamate surge that happens from these ionic changes. When it's excessive, it can kill cells. Really what happens is you get a metabolic crisis because the neurons deplete all of their ATP and intracellular energy stores, can't maintain the sodium-potassium pump. And then that triggers the increased calcium, triggers calpains and caspases. Reactive oxygen species form, mitochondria become impaired, and then apoptosis is the end. Just to remind you, Wallerian degeneration, also unique to the CNS, when these fibers get crushed or severed, there's, the distal neuron, the distal axon degenerates. And so this is kind of like an atypical cell death mechanism, since the neuron itself that it was connected to that axon doesn't die, can stay alive, but potentially over the long term, when you have a neuron that gets disconnected and it's not receiving the growth factors that it needs from its downstream target, that can lead to problems as well, potentially. So here is an immunohistochemical stain showing these spheroids in a diffuse axonal injury context. This is from Johnston et al., 2013. So what you're going to see are these axons and the white matter with these swellings. You might see kind of a beaded appearance. This is APP, immunohistochemistry. So this is an extremely useful stain, the amyloid precursor protein. It's the same protein that is involved with Alzheimer's disease, where the A beta peptide comes from. But it's just an example of a protein that is transported by fast axonal transport. So it really very nicely brings out diffuse axonal injury. I just thought it was super interesting that other factors that are important for neurodegeneration, including the beta secretase, presenilin 1, and the A beta peptide, are also going to be positive in these spheroids. So this is a potential link. More work needs to be done here, but maybe a potential link between short acute TBI and long-term TBI. All right. So now it is 12:27. We are halfway through the talk and halfway through the hour. I'm going to take this opportunity to have a sip of my Diet Coke, which I'm addicted to. All right. So chronic traumatic encephalopathy and the tauopathies. So I don't know what you all think, but to me, the tauopathies are probably among the most interesting brain diseases. There's so many different contexts and so many different mechanisms. You're going to have cognitive disorders, movement disorders, motor neuron, and psychiatric symptoms in patients with tauopathy. So pretty much touches everything that can go wrong with the brain. The neuroanatomy is super interesting. The neocortex is involved in some, subcortical, brainstem regions, even the spinal cord in some. So the neuroanatomy variably affects different parts of the brain. Histologically, you can see tau in neurons and astrocytes and oligodendrocytes. Never seen tau in a microglial cell or an ependymal cell, but still really interesting. And the relative amounts of tau in the different types of brain cells is going to really help you with diagnosis. There's also a whole biochemical and ultrastructural component with different tau isoforms with different numbers of microtubule binding domain repeats, different modifications, phosphorylation, and filament ultrastructure that I'm going to touch on that are different but overlap. And then there are these mutational cases, people who have mutations in the tau gene, autosomal dominant, that cause frontotemporal dementia. But there's also common risk alleles. There's this 17q2131 haplotype, which is among the most complex structural rearrangements in the human genome. But it's a hot spot for neurodevelopmental abnormalities, and there's a lot going there from a cytogenetic perspective. What are the different primary tauopathies? So Alzheimer's disease is thought of as like a secondary tauopathy, with A beta being the kind of driver. These are diseases where tau seems to be the main thing. So the nomenclature is a little confusing. There's this category of frontotemporal dementia with Parkinsonism linked to chromosome 17, or frontotemporal dementia with a tau mutation. Those are really rare. There's a lot of more common things. I've spent a lot of my time working on primary age-related tauopathy, which is extremely common, but only dementing in a small minority. There's the age-related tauopathy of astroglia, PSP, and corticobasal, which are both Parkinsonisms. There's the Grain disease, Pick's disease. There's anyway a lot to look at. But I'm going to just point out that many of these diseases are characterized by they're sporadic, and they're characterized by an association with the tau inversion. So genetics, but chronic traumatic encephalopathy is environmental. It's really thought to be caused, and I'll explain more about that, that the data is irrefutable at this point, caused by an environmental insult. So it's a interesting context to compare risk and environmental risk and genetic risk, and modeling that scientifically is also fascinating. Some of you are probably interested in brain tumors. Some gangliogliomas actually get neurofibrillary tangles in them. If you come across one, let me know, because I'm always on the hunt for gangliogliomas with neurofibrillary tangles in them. If you could share them for research, that would be amazing. But there's a huge opportunity there. And then there are viruses that can cause tangles as well. So you pretty much have a lot of different contexts to kind of compare. But not every brain disease has tangles. And so why is it these diseases specifically, but not others? Like, you don't get tangles after a stroke. You don't get tangles in multiple sclerosis. So there's something that links these diseases. I don't know what they are. If you know what it is, let me know. Or even better, figure it out, write a paper, win the Nobel Prize, and tell them that I inspired you. All right, so these are some examples of some of the tau structures that we see. These are all tau immunohistochemical stains with the AT8 antibody. So these are examples of neuronal tau structures. Here's some glial tau structures. There's the neuritic amyloid plaque, which is a large extracellular structure, and you're going to see the tau positivity and the dystrophic neurites around it. Here's a classic neurofibrillary tangle, which is an intracellular structure that has a flame shape. Sometimes they form these little circles, in which case it's going to be a pick body. There's this kind of like eyeliner cell where you get a nuclear ring of tau positivity. You can get these granular kind of cytoplasmic structures. You can get grains. These are the grains, examples of these dot-like structures in the neuropil. Here are some glial tangles. This is a tufted astrocyte in PSP, which looks very different from an astrocytic plaque in corticobasal degeneration. Really, it's like the tau in the PSP one is in the cytoplasm, the somato compartment of the astrocyte. And in corticobasal, the tau seems to accumulate in the distal processes of the astrocyte. Nobody understands why that is different. Here's an example of a coiled body. How and why tau gets into oligodendrocytes is unknown, but it's just been shown, or people have hypothesized this before, that oligodendrocytes can actually be phagocytic, and they may be phagocytizing tau that's positive in abnormal axons from neurons. And here's an example of a thorn astrocyte that can be seen in age-related tau astrogliopathy. So a little bit more about tau. This is a drawing trying to show that not only does tau progress through the brain in a hierarchical progression, there's also a sequence of events, sequence of tau pathology, where you may have a normal neuron, and the first change is these kind of granular tau oligomers that can maybe spread from cell to cell through extracellular vesicles or other means. These little pre-tangle structures eventually coalesce to form an intracellular tangle, pushes the nucleus to the side, and you can see that under the microscope. It's a sign that you have a strong space-occupying lesion, and then eventually that cell will die. Normally, tau, we think of it as a microtubule-associated protein that stabilizes microtubules, probably plays a role in axonal transport and heart outgrowth, maybe growth factor signaling. We really don't know exactly what tau does, which is a little bit embarrassing because I've been working on this since 1997. I still haven't figured it out, but we still don't know what A beta does or alpha-synuclein. So it's not just me. The toxic tau protein, we also don't really know exactly how it kills cells or even how the tau protein is related to A beta. Post-translational modifications seem to play a role where phosphorylation of tau maybe transforms it into a toxic structure. Neuroinflammation, oxidative stress, all the other usual suspects have all kind of been implicated. Many of you have done a lot of work on tumors, and you may have noticed that the neuro-oncologists are way light years ahead of the neurodegenerative disease pathologists in terms of understanding mechanisms. It's a fact. I think this is a harder question to answer. If you're wondering why we're so far behind, I think it's because when you're dealing with cancer, they grow in a dish. The neurosurgeons give you lots of cells to play with. They're cell autonomous functions. These are non-cell autonomous processes that involve a lot of different cells in coordination with one another. So that's one of the big barriers that we have to deal with. Anyway, this is a schematic showing kind of an overview of these tau mechanisms that you need to think about. Mutations in the tau locus, or these common variations of tau haplotypes that I mentioned before, the 17q2131 inversion. The tau gene gives rise to different mRNA isoforms. There are 6 of them, primarily in the brain. They can have 3 or 4 microtubule binding domain repeats. Just to remind you, some of the tauopathies have more 3R tau, some have more 4R tau. The classic 3R tau tauopathy is Pick's disease. Most of the rest of the rare ones are 4R tauopathies, PSP, corticobasal, AGD, and globular glial tauopathy. The common ones, AD and PART, are mixed, as is chronic traumatic encephalopathy.
and some of the FTLDs. The tau mutations are funny because depending on where the mutation is, they influence splicing in different ways. I mentioned this before, the tau becomes phosphorylated, it aggregates, and then the cells die. But how this process happens is unclear, and which are the most important steps, and how tau can propagate, like a prion potentially, is also important. What's really interesting to me is that there are some contexts where tau becomes phosphorylated in the absence of pathology. There's an amazing paper that one of my trainees, Marco Hefti, did looking at tau in human brain and neurodevelopment. There's a tremendous amount of tau phosphorylation in a normal human fetal brain, and even tau aggregates forming. That can also happen during hibernation of hibernating animals. So why do we get hyperphosphorylated tau aggregates in a normal human fetal brain? Nobody really knows. This is something that I'm hoping one of you out there will go out and study and give us the answer. But what about CTE? That's why you guys are here. So this was first kind of picked up on by a guy named Martland, Harrison Martland. It's kind of depicted here. He didn't actually get all of the pieces together. What he noticed was that there was a subgroup of boxers that he described. He said they were punch drunk, and that was kind of the slang term for them. They call them they had other terms for them. If you read this paper, it's actually kind of entertaining. But he really focused on the vascular component and the hemorrhage. He didn't actually realize that there were tangles and also plaques. Later, the term was kind of, the entity was renamed dementia pugilistica. I don't know why dementia pugilistica. Maybe give it a Greek name and it sounds more profound. And then they switched it to chronic traumatic encephalopathy. That was just to recognize that it wasn't just the pugilists, the boxers that were getting it. It was pretty much anybody who had repetitive mild traumatic brain injury. And that can happen in a lot of different contexts. So, for example, any combat sport where you're getting hit in the head, intimate partner violence, blast exposure. There were autistic children who had a headbanging behavior, and they all had the same kind of pattern. The thing that linked them together was mild yet repetitive concussive, and there's also a term now, subconcussive injury. So these are people who don't have a lot of symptoms, but still maybe have the mechanical injury to the brain parenchyma. What are the symptoms of CTE? Well, it's pretty much everything. The younger patients get more psychiatric stuff, and the older patients have more cognitive and movement disorder sorts of things. So it can progress to movement disorder, motor neuron disease, and so on. There's one little aside. I think it's really interesting that many of the kind of poster children for different degenerative diseases likely had traumatic brain injury induced neurodegeneration. So Muhammad Ali was a boxer. We all know that he had Parkinsonism. Many feel that he probably had a traumatic Parkinsonism, not a classic garden variety GBA LRRK2 mutation. There was no autopsy, unfortunately, so we don't really know. Lou Gehrig, ALS, amyotrophic lateral sclerosis. He had 9 documented concussions when he was in the major league. He also played football when he was at Columbia University. He was a very aggressive player. He may not have had Lou Gehrig's disease, which is ironic. It's possible, and this is in the literature, you can read about that. It's possible that he had a chronic traumatic encephalomyelopathy. No autopsy was ever performed, so we'll never really know. Even Ronald Reagan, who had Alzheimer's disease, if you read Nancy Reagan's biography, she says that his cognitive symptoms started when he fell off a horse. So he may have had a traumatic brain injury-induced Alzheimer's type disease. And we know there's lots of examples of patients who have been diagnosed with Alzheimer's disease who are in our brain banks, and then we've gone back and screened them for CTE and found that they have the telltale signs of traumatic brain injury-induced neurodegeneration that I'm going to show you. So I think there's a lot of work that needs to be done to better define these diseases while people are alive, and the autopsy studies are the key to doing that. Okay. So gross pathology, gross neuropathology, super important. So what do you see in CTE? There's nothing specific, but there's a lot of suggestive features. Atrophy and ventricular dilatation, especially in the later stages. The septum pellucidum can split. So you may see a cavum, you may see fenestrations of the septum pellucidum. So if you get a case and you're suspecting CTE, take a close look at and document any changes in the septum pellucidum. The mammillary bodies and other brainstem structures can be involved, which is not really classic in Alzheimer's disease, although you can have some. The substantia nigra and the locus ceruleus can have pallor, and the corpus callosum can be thin. Any projection nucleus, any place where there are long axons, you're going to have vulnerability to mild repetitive traumatic brain injury. What are you going to see under the microscope? There's a lot of features that have been described. The first one, these are some tau stains. The first is going to be a sulcal predominance of neurofibrillary degeneration. So why the sulcus? That's an area of stress concentration. It's just biomechanically a place where energy gets dissipated as it permeates through the skull. And it has to do with geometry and the material properties of the brain. I kind of think of like a Kit Kat bar, bar of chocolate, and I love chocolate. Break the chocolate bar, it doesn't crack at the top, it always cracks at the bottom because that's where it's weakest. So this is kind of as the brain moves, this is where it's going to be weakest. Another thing that's different is in Alzheimer's disease, you're going to see a lot of tau tangles in the deeper layers of the cortex as well as the superficial layers. In CTE, it's not all or none, but you're going to see a predominance at the superficial layers 2 and 3, superficial layers. It's also going to be patchy in CTE, which you can kind of see in this first panel here. Another thing that you're going to see are grains. The grains in CTE, we don't really fully understand. Sometimes they're referred to as dot-like structures, grain-like structures. They're irregular and they're larger. We don't know how or why those form, but they might be related to shearing of neurites. This is the most important thing to understand. It's probably the one thing that you should walk away from this presentation with is how to recognize a pathognomonic diagnostic lesion of CTE. So it only happens in CTE. We don't know why. Sorry, we do know why. We know exactly why, because it's biomechanical, that you get these perivascular neurofibrillary tangles at the depths of the sulci. So these are some pictures from cases in my collection. This is the, let me describe what we're seeing here. I'm starting in the middle because it's maybe one of the most spectacular examples that I have. This is the peel surface here that I'm tracing with my mouse. There's going to be these thorn astrocytes. This is R-tag, kind of deep in that sulcus. This is suggestive, but it's not the feature that we're looking for. The feature that we're looking for is neuronal perivascular tangles. So we see this a lot in aging and other contexts. But as you follow these sulcal vessels down, you're going to find collections of neurofibrillary tangles around them deep in the cortex. Not in the white matter. It has to be in the cortex. It has to be neurons. Astrocytes might be there, but neurons are the key feature. Here's an example of a blood vessel here with the target, the neurons around it forming that pathognomonic lesion. There's not that many things in neuropathology that are pathognomonic. So this really stands out among the Tauopathies. I think the tufted astrocyte is essentially pathognomonic of PSP. This is kind of essentially, it is pathognomonic of CTE. The reason why this happens is it's also a biomechanical phenomenon. Stress gets released at interfaces between different materials, especially when they are different, they have different elastic properties, different rigidity. So I tell people to imagine a bowl of Jell-O, okay? And that's a brain, and you can shake that bowl of Jell-O, and it's going to do just fine. But if you bake something into your Jell-O, now imagine you've baked in a knife, and you shake it, you're going to have shearing and tearing of stuff all around that structure. So turns out that blood vessels are much, much more rigid than the brain parenchyma around them. So right around that blood vessel, when the brain is getting moved, it's almost like, I don't know, for all you people who are into pottery out there, it's kind of like having a wire in your clay. You start shaking that clay, you're going to get little holes in it. You're tearing the brain with these vessels. I hope that makes sense. Anyway, you need to be able to recognize the difference between an astrocyte and a neuron on a tau immunohistochemical stain. The neurons look like neurons. They have dendrites and axons and the somatodendritic compartment. Astrocytes in general look more fuzzy. They have irregular borders and stuff. Anyway, there's the pathognomonic lesion. And all of this stuff can be modeled, which is really, really interesting. They can build these finite element models, which are things that engineers do when they're trying to looking at uh, the strength of different buildings or planes or whatever they're making. This is a famous paper at this point, Gajari et al., 2017, when they modeled the forces getting applied to a brain, and they found that there's a massive increase in the amount of force that gets released at these sulcal depths, and that can be seen here in these images. I think that one thing that's kind of interesting to point out on this presentation of this paper is that everybody's brain is slightly different. Some people have larger brains, some have smaller brains, some have longer brains, and not all of our gyri and sulci are exactly the same. The tentorium of our cerebellum is not always exactly the same. So depending on the shape of your head, you're going to be more or less vulnerable to TBI in different brain regions. I think that's a really exciting and interesting idea, because what it means is you could potentially get a scan before you participate in any activity and get a sense of whether or not you're preferentially vulnerable or not. Another thing that's been really interesting and maybe confusing about CTE is that it's really common to have mixed pathology. So in neurodegeneration, it's becoming increasingly obvious. We always kind of knew it, but now it's becoming more of an urgent problem because of biomarkers and categories, that polyproteinopathy is more of the rule than it is the exception. People will have many degenerative proteins, markers at the same time, and that can lead to a lot of confusion. So CTE is no exception. Probably the most common comorbid, well, I guess amyloid is going to be the most common. Patients that have CTE are going to have some plaques and some diffuse and neuritic plaques. They're going to happen younger, and they're going to be more severe than compared to classic Alzheimer's case. There's also increases in cerebral amyloid angiopathy that I'm not talking about. TDP-43 is also extremely common. Alpha-synuclein, for some reason, not that common in these CTE cases. So really, TDP-43 and amyloid are the most important. And if you're interested in reading more, take a look at Dr. Torstein's paper in Acta Neuropathologica, and he's got many more where he's been characterizing these comorbid pathologies and trying to understand what it means. You know, when you think about this, it can kind of trigger a little bit of an existential crisis. Does this mean that they're just getting two diseases, or is TBI kind of bringing out a risk for a disease that was already there? We need to do a lot more work here, but it's really important. This is a critical kind of must-read paper, the consensus criteria. This is the first consensus criteria for CTE. There's a second consensus paper that followed this. It focuses on other things, sampling and staging. This is the basis of our criteria at this point. So the first thing that's absolutely required for a diagnosis of definite CTE is having the pathognomonic lesion. These are the supportive features. I mentioned the superficial tangles. There's also more in CTE, and I'm going to show you a little bit more. There's more tau in CA2. Alzheimer's disease, classic Alzheimer's disease, CA2 is protected, believe it or not. So there's a different hippocampal kind of fingerprint of tau pathology in CTE. You're also going to find subcortical nuclei getting involved. You're going to see these thorny astrocytes at the glial limitans. I was showing you that at the depth of the neocortical sulcus. You're going to see these grains and dot-like structures. I mentioned these growth changes before, and TDP43 positive inclusions are also suggestive. So here's the staging system for CTE on tau pathology that was proposed by Anne McKee in 2013. In CTE, the very, very first thing that you'll see are these depths of the sulcus tau deposits. Then they slowly become, they kind of bloom, they start to spread out, they start to move from the sulcus out to the side, involve more sulci. In the later stages, the medial temporal lobe gets involved, and then it becomes confluent throughout the whole brain. It's really interesting conceptually that it's the opposite of the Braak staging. So Heiko-Braak for tau and aging and Alzheimer's, it starts in the hippocampus and then spreads to the neocortex. This disease starts in the neocortex and then spreads to the hippocampus. There's been a lot of controversy around CTE, and I'm still really surprised that the controversy continues. There's a lot riding, a lot at stake. There's a lot of people who are involved with organized sports and football, so I can imagine why the emotions might run high. But you're still going to find people who are doubting that CTE is even really a real disease. And so this is just one example of an article in the Star Tribune from 2018, citing a paper by Ling et al. Saying that CTE changes are present in 12% of normal people, and saying that the evidence is not strong, causation has yet to be settled. This is not true, and I'm going to explain why. If you look at the Ling et al. Paper, they describe these perivascular lesions. These are the astrocyte lesions that we see in aging-related tau astrogliopathy. These are thorn astrocytes, fuzzy astrocytes in the white matter. The CTE pathognomonic lesion is neuronal, and it's in the gray matter. So please, 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 when you're looking at your cases, remember that the pathognomonic lesion is neuronal and it's in the gray matter. You may see astrocytes in the gray matter, but these perivascular lesions of the thorn astrocytes are in the white matter, and they are common in aging, and they're not the CTE lesion. Here are some examples of thorn astrocytes. You can see they're stubby. This is a neuron like next to it, and they have these short little fat processes. Here's an example of a granular fuzzy age-related astrocyte, which can be found in the cortex. The concept of RTAG, aging-related tau astrogliopathy, was really codified by Gabor Kovacs, and these are our consensus criteria for that. You'll often hear people say that CTE is just normal aging. We've spent a lot of time looking at age-related tauopathy that happens in the hippocampus. This is also required reading. You probably have made this diagnosis many times because you've seen it in essentially every single case over the age of 50. So there's a pattern of tau pathology that occurs in the hippocampus as you age. It's different from CTE also. There is a little confusing thing here. In primary age-related tauopathy, you get a lot of tau, often in CA2 and CA1, as you also do in CTE. The difference is in CTE that there's a lot of tau also in CA4 and CA3. So that can kind of help you. Basically, the tau pathology in part is mostly CA2 and CA1. You're not going to see as much in other regions. CTE, when it's florid, the tau is essentially everywhere. If you're talking about early CTE, things can get a little bit more complicated. I'm going to end here with this age prediction model. Maybe I'll rush through it. So we've been doing a lot of AI in the lab. If you're not doing digital pathology, and if you're not starting to bone up on digital pathology, get started. It's going to be transforming our field. Being able to measure age acceleration is a huge thing in research. So, and it's going to be important clinically, I think, as well. We trained an AI model. This was Gabe Marks, who's an incredibly talented master's student, now a research track neurology resident, that can predict an age based of a person based on their hippocampus. You just show them the hippocampus, and it tells you how old that patient is. and it gets it within 5 years. This is an extremely powerful tool because it allows us to look at the difference between the predicted and the actual age, get a measure of age acceleration, and then we can study that age acceleration and see if there are genetic factors and environmental factors that are doing this. And the way that the AI does it is it doesn't look at one feature, it looks at every feature that it can extract from the brain. Thousands and thousands of things, cells and structures and things that are perceptible, subtle, or imperceptible to the human brain. But it does that by running it through a giant neural network that can pull out thousands of features. This is essentially the same neural network that the self-driving cars use, except applied to the brain, and you can train the AI on it. And then the AI will tell you which brain regions are responsible for the accelerated aging by putting this heat map on top of it. This is really dramatic. The football players that we have in our cohort, they have a dramatically accelerated aging phenotype. They're predicted to be over 10 years older on average than people that didn't participate in sports. So this is a completely unbiased measure of what we think of as a severe age acceleration in CTE. Just to finish up with the controversy, CTE is extremely strongly correlated with participation in contact sports. There's many papers that have done this, and the p-values are extremely, extremely low. Take a look at this paper by Nowinski et al., applying the Bradford Hill criteria to CTE and mild TBI. Bradford Hill criteria are an epidemiological system that was used to essentially establish causality between smoking and lung cancer. Lots of things are important: consistency, the strength of the association, how specific it is, the temporal gradient, or sorry, temporality, the environmental insult, and then the effect, dose response, it's plausible. Basically, CTE has satisfied all the criteria set forth by Bradford in 1965. So as far as many of us are concerned, TBI, mild repetitive TBI, causes CTE. Some scientists disagree, but that's their fault. So I'm going to skip this little bit on the tau isoforms. Maybe I just tell you that the tau isoforms in CTE are thought to be the same as Alzheimer's disease. However, there is something that's a little different. It seems that they start more with 4R tau, and then the 3R tau kind of comes in later. And the ultrastructural experiments, really interesting. There are different ultrastructures of tau filaments. The CTE fold is different, and you can look at these cryo-EM structures. And there you go. And I finished with 1 minute left.
Yes, you did. Thank you so much. Unfortunately, there's very little time for questions. If at all, can I suggest that people email you? Would that be okay?
You are perfectly welcome to email me. Yeah, I can stick around for another 5 minutes if anybody wants to stay on, if the Zoom is open.
All right. I learned a lot. I really appreciate your explanations. And I have an incredible case of a ganglioma with neurofibrillary tangles, but I don't think I have any tissue left. So I'm sorry about that. I guess there's
Actually, if you have slides, what I'm really interested in is the genetics.
Yeah,
I think
I
those
have some.
studying the genetics, if they have rearrangements in the tau locus, or do they have other factors that are driving tau formation, tangle formation? Really interesting.
Interesting. There is a question in the chat. Do you want me to read it?
Yeah.
Yeah. So Elena Dowd asks, do you find it virtually impossible to identify low ADNC in the background of high CTE, and vice versa? With high ADNC, is there a significant pitfall of missing CTE? Any helpful tips to address this during diagnostic workup? Does the high tau burden in CA4, CA3 also apply to ADNC?
So yes, it's such a hard problem. It's a bigger problem when you have somebody who has high Alzheimer's changes, and you're like, is there CTE lurking in here? So the first thing that I would recommend is go to the heterotypic neocortical areas. Go to the occipital cortex, go to primary motor, because they'll have the lowest amount of tau. See, the big problem is when there's amyloid there, it blots out the sky. it's like spilling ink all over your brain, and you're not going to see the CTE lesion. So that's one tip. Go to these regions that are least involved. But if you have a very, very severe Alzheimer's, you might not even see it. Then we thought, go to the brain stem. And then if you see stuff in the mammillary bodies and these other regions that you don't typically see in classic Alzheimer's disease, that might be a suggestive feature that might help. Another thing that can help is if you want to move to some isoform specific antibodies. If you try the 3R and the 4R, that might be able to help you bring out kind of that sulcal predominance that you might see in CTE. If you have a patient that has like a strong history of, say they're a prize fighter, and they've been knocked out and they have AD, you have to maintain that suspicion no matter what. We're not good at getting all of the features. There's a lot more than just tau, right? So there's going to be more papers, there's going to be more studies, we're going to identify more signatures in the future. So just, keep it open that some of these cases, just because you can't find the pathognomonic lesion, it's not CTE. And then what was the other one? The other way around. Yeah. So if you have some mild Alzheimer's changes in somebody who has CTE, you kind of have an existential crisis. You don't know if that Alzheimer's change is just kind of going along for the ride with the CTE, or if it's really an independent pathological process. We don't have a marker that can say this is if you see a neurofibrillary tangle in CA1, you can send it with AT8 and say that's a tangle, but I don't have a marker that's specific for Alzheimer's tangles versus CTE tangles versus PART tangles. What you can use is neuroanatomy, right? If you have a CA2 predominance, you're going to say, okay, those tangles in CA2 may be CTE, they may be PART. Classic Alzheimer's doesn't really affect CA2 very much, but you can get affected a little bit. So we need to do more work here. I really think the spatial transcriptomics, spatial proteomic studies, hopefully, if we're lucky, will give us some specific markers, but we don't really have them yet.
You're almost getting me excited to go back to doing research in neurodegenerative diseases, which I haven't done for a little while. Okay, I'm not hearing Sarah. I think the information is in the chat.
Yep, I can go ahead and do closing announcements. Sounds good. Well, thank you everyone for staying on just a little bit later, and thank you again for joining today's AANP Teaching Rounds presentation. We would ask that you take a few minutes to complete a short evaluation. Completion helps ensure accurate reporting to the accreditation board. A link to the evaluation will appear on the Path LMS screen upon your conclusion of the Zoom meeting, or you can navigate back to the main course and select the evaluation option. Once you've completed the evaluation and selected your credit amount, please then select the appropriate certificate based on your credentials. And then the PowerPoint slides and recording will be posted to the AANP website in the next week. And thank you again to Dr. Crary for an excellent presentation. And that concludes the session for today. Thank you so much, everyone.
Thank you. And you should know we had 66 participants most of the time.
66, wow.
Yeah. All
And
right.
some of them actually stayed on. Thanks so much.
That's correct. Thank you.
Thanks again, everyone. Have a good day. Bye.
Bye.